All right, so far in class we've talked about where cells, how we first learned about cells. We talked about the barrier around cells, the cell membrane, and kind of how it separates itself, how a cell is separation from the outside world. Then we talked about what's in that separated compartment. So how does that separated compartment turn into something that can go through all the processes for life? And then next part, what we're going to talk about during this section is how do cells make new cells? How do you take that living, either that single-celled organism that's a living thing, or that component of a living thing, that part of a living thing, and make new parts so that either if it's an organ or a tissue, so that it can go on living and then continue functioning. So the title for this section is Cell Division. So first you ask, why would a cell divide? Why not just have a single cell or cells that just keep going on forever? Cells are just like, oh, I'm glad that happened at the very beginning. Cells are just like, your vacuum cleaner or washing machine. Parts of them break down or they stop functioning. And so you need a way, once it stops functioning, to create a replacement. So one of the reasons cells divide is reproduction. Cells are going to be making new cells. If it's a bacteria like this one over here, it's a rod-shaped bacteria. For it to create offspring or to create some sort of copy of itself to continue on, to allow its DNA to continue on, to continue living, it divides for reproduction. Other reason is for growth. We talked kind of very early on about how Pasha, well, in one class we talked that Pasha is a different size than a blue whale. Not because Pasha has smaller cells, but because he's made up of less cells than the blue whale is. So the baby has less cells than, as the child continues to get older, it adds on cells and gets bigger. And the other part is for repair. So if you injure yourself, those cells might get damaged and never, they can't be used again. So repair is another big part of why cells would divide. We got to create healthy cells to cover up the area that was damaged. So how does a cell know how to divide? So why? Why would it just start dividing, or how does it control that process? That's going back to the nucleus. So we talked about the nucleus as if it was a part of the city center. Maybe it's the mayor's office. DNA has the instructions for all the cell's activities, including cell division. So the DNA is going to cue the cell in for, okay, it's time for this process to start now. And it would cue in the time for cell division. And here's a picture basically breaking down the different sequences of development and division, the steps that happen in the cell cycle. It's called cell cycle or mitosis. It has three main parts: interphase, and if you look at that pie chart, interphase is a huge part. That's just the preparation. It's the cell tooling up and getting ready, everything it needs before it divides. Mitosis is the actual steps of cell division. So if you look here, mitosis is outlined in this wedge, and it's composed of these steps. And then the very last part is cytokinesis. Cyto is cell, kinesis means splitting, and so that's the full splitting of the cell. So the mitosis is the nucleus of the cell dividing. Cytokinesis is the full cell, the whole thing dividing. And at the very end of cytokinesis, you have two cells. So adult human cell takes about 24 hours for that to happen, for it to divide. And if we'd looked at this pie chart, 18 to 20 hours then would be that interphase, that tooling up of the cell. 2 hours in mitosis. So if you wondered, how do you go from a single cell just formed in a mom to then a baby coming out, a few months later? Embryonic cells divide every 30 minutes. So that's how you're able to go from just that single cell to a cluster of cells and then eventually form a tiny baby, because it happens a lot more rapidly. If it was once every 24 hours, like a human cell, it would take a really long time to form a baby. So let's look at these steps. Let's break them down. Interphase. So I want you to know the step and then what happens. So as the cell's preparing, it doubles in size. DNA replicates, so you can see our little DNA strand doubled here. Organelles double in number, so here we have our centrioles. They double. All the organelles double in number. And that's because, imagine splitting the cell, doing everything you need to split the cell, and you have one cell that has all the functional organelles, and one cell that's just a lipid membrane and some DNA. You need to make sure you have all the functioning parts in both cells that split off. Okay, next step is prophase. DNA forms chromosomes, so it kind of looked like angel hair pasta before, and now it's kind of condensed. It looks like farfalle, like a bow tie pasta. The DNA, the name for that structure that forms, the condensed DNA, is a chromosome. And you've probably heard of chromosomes, you've probably heard of DNA before. Chromosomes are made up of DNA, they're just the condensed version. Spindle forms, I don't have a nice animation for this one. So basically the spindle is like this set of it's almost like railroad tracks, these bars that go across, and it's something to attach the DNA to, so the DNA can be split and moved across to the part of the cell that it needs to go to. So that's the spindle apparatus. And then the centrioles move to opposite poles, and they're the thing that's going to kind of operate that spindle and kind of move move things along on the spindle. So those are the three things for prophase. Oh, and then also the nuclear membrane begins to disappear. So we've condensed the chromosomes, or we've doubled the DNA, we've condensed the DNA, and eventually we're going to need to move it. But if we have that nuclear membrane barrier, it'd be really difficult to move the DNA around. So that nuclear membrane has got to start to disappear so we can move the DNA. So next we have metaphase. The nucleus has completely disappeared at this point. That means the chromosomes are free to move around. The chromosomes are going to line up in the middle of the cell. We call it the cell equator. So the cell is round. You could imagine if it was like the Earth, it has an equator down the center. So at the center, we're going to line those chromosomes up. And another way, just like a way to remember what happens at metaphase, metaphase starts with M, middle also starts with M. That's the biggest part of what's happening at metaphase, is those chromosomes lining up in the middle. And then next we have anaphase. So what happens? Centromeres divide. So basically what happens is we had those, that chromosome, and each, it looked like a butterfly. So part of the chromosome was here, part of the chromosome was here. It's held together by a thing called a centromere. That centromere divides, which allows you to take half of the DNA information, or half of the chromosome, over to one side, and half of the chromosome to other way. We call the little segment of the chromosome a chromatid. It separates and moves to opposite poles. So if we look at this phase, just like we did with metaphase, anaphase starts with an A. The word away also starts with A. So what happens during anaphase is the chromosomes move away. And then the two new vocab words we introduced on this slide are centromere. Centromere is the thing at the center that held together our chromosome. And a chromatid. A chromatid is just half of a chromosome. It's this part. Each one of these is a chromatid. Next up is telophase. The nuclear membrane forms around each group of chromosomes. So we're reforming a nucleus, and the chromosomes then can unwind and go back into their little angel hair pasta form. And cytokinesis begins. So this area here is called a cleavage furrow. It's where the DNA, or it's where the cell starts to split and starts to pinch in. So it's just beginning here. It's just beginning to pinch in. If you think back to the very beginning of this presentation, I said what cytokinesis is. Cyto is cell, and kinesis is division. So cell division begins at this step. For cytokinesis, we now look, you've got two fully formed cells. So cytoplasm divided. Cytoplasm, again, was the thing outside of your cell. One cell has become two cells, and they're fully formed. And that's the very last part. There's a lot of new science vocabulary and new words. If you were listening to the slide, then listening as I presented it, that's going to be helpful. If you just read it and wrote down, probably not going to remember those words. But look at the pictures visually, get an idea of what they look like, and familiarize yourself as you watch the video. So then as you try to learn that word, it's a long word. It's a science vocabulary word, so a little bit tricky. But look at the picture first, get a good understanding of what it looks like, and then start to work on pronunciation of the word and work on that definition of what the word is. Good luck, and we'll see you back in class.